Hello everyone. My name is Dr. Munmi Bora, plant virologist, Department of Plant Pathology, Assam Agricultural University, Jorhat, Assam, India. In this lecture, we will be discussing about Introductory Plant Virology, and this lecture is specifically for the students. So let's move to our main topic. Viruses are group of acellular infectious agent. Acellular meaning containing no cells and whole of the particle is called virion. Viruses consist of nucleocapsid. That means nucleic acid surrounded by a capsid or coat protein. Here we can see criteria of being alive not fulfilled by a virus. In this table, we are comparing virus with a bacterial cell. You can see viruses do not perform metabolism, they do not have capacity for reproduction, they do not respond to stimuli, they do not have any cellular composition, and they do not have complex growth. So biologically, viruses are inert and not possessing any biological function. Viruses actually lack the protein synthesizing machinery and it depends on host metabolism for their growth and multiplication. There are some terms associated with viruses: obligate, that means cannot be cultured inside laboratory like bacteria and fungi. Submicroscopic, cannot be visualized using light microscope. Intracellular parasite, parasite inside the cells of the host. Genome is either DNA or RNA, never both present in a particular virus, and the genome is surrounded by a protein coat or capsid. So there are lots of curiosity regarding viruses are living or non-living. We could see that viruses do not fulfill the criteria for being alive. But inside the host organism, we can call them microorganism. But of course, outside of the host, they are just molecules or particles. So viruses are a system of intermediate complexity in between biomolecules and living organisms like bacteria. Viruses are actually considered as self-replicating molecules and they depend on host system for this replication process. You see, even though not fulfilling criteria for being alive, viruses can cause disease in plants. How? Viruses enters into the cells through wound, that is any mechanical injury, and replicates inside the cells, moves to neighboring cells locally through plasmodesmata, and then systemically to the entire plant by vascular bundles, that is the phloem cells. In this picture, we can see cell-to-cell movement of viruses takes place through plasmodesmata very clearly and long-distance transport through phloem tissue. Plant viruses express movement proteins. In this picture, it is marked in red color, which alter the plasmodesmata. This is one of the mechanism of cell-to-cell movement. So symptoms due to virus infection may be necrosis of cells, hypoplasia or hyperplasia. You see the lower picture, localized necrotic flares 2 days after inoculation. That is local symptoms. And after 2 weeks, systemic necrosis occurs and ultimately plant dies. This is a kind of systemic symptoms. Regarding the size of the viruses, of course, it is submicroscopic. The largest smallpox virus is more than 5 times smaller than E. coli and 50 times smaller than human RBC. Size of the virion may ranges from 10 to 300 nanometer in diameter, and this is for your information that the longest plant virus is Citrus disteza virus, size of which is 11 into 2000 nanometer. So, do you believe viruses are molecular hijackers? Yes, because once viral genetic material inserted to host genome, it hijacks the host cell machinery to make copies of the virus. In other words, it forces the host cell to produce more numbers of virion. We need to understand here that viruses are specific to the kind of cells they infect, and this specificity depends on the types of receptor present in the host surface and the receptors present over the surface of the virus. So coming to the definition of plant viruses, Matthew 1981, he described viruses are entities whose genome is an element of nucleic acid, either DNA or RNA, which reproduce inside the living cells and uses their synthetic machinery to produce specialized particles called virions, which contain the viral genome and transfers to other cells. In a broad sense, therefore, we have only two groups of viruses, RNA viruses and DNA viruses. Latin binomials were proposed first by Holmes in 1939. We will not go into details of this taxonomy of viruses. Currently, two main schemes are used for classification of viruses. The International Committee on Taxonomy of Viruses, that is ICTV system, and Baltimore classification system. Baltimore classification was first defined in 1971, which places viruses into 7 groups based on their nucleic acid types. There are some other definitions given by different scientists, so this is for your information. So please go through it. Coming to the history of virology, the term virus was originated from Latin word which means poison. Report of tulip color breaking is the oldest known example of virus disease. It was reported in the year 1576 by Carolus Clusius. Actually, in those days, kings and queens of Netherlands were very much crazy about a kind of variegated patterned tulips, now known to be caused by tulip breaking virus. This virus induces in foliage of infected plants beautiful variegated color patterns, and such bulbs were extremely expensive, giving rise to tulipomania in the 17th century. In 1886, Adolf Meyer was a professor at Agricultural College Wageningen, Netherlands, observed highly contagious disease of tobacco which could infect healthy plants by injecting diseased leaf extract. He gave the term mosaic krankheit for mosaic disease of tobacco. In 1884, the French microbiologist Charles Chamberland invented a filter which could pass solution containing bacteria and completely remove them from the solution. In 1892, Dmitry Ivanovsky confirmed the findings of Mayer, also observed that sap of diseased plant remained transmissible even after filtering it through the Chamberlain process and factorial filter. He doubted toxin produced by the bacterium may be the cause of mosaic disease of tobacco. And after that, in 1898, M.W. Beijering in Netherlands finally established the cause of tobacco mosaic disease and named the agent as Contagium vivum fluidum, and he gave the term virus. He was called the father of virology. Now, transmission of plant viruses. Transmission behavior of different viruses by their vectors vary considerably. Broadly, it is divided as horizontal and vertical transmission. Horizontal means transmission by mechanical means, and vertical means transmission through propagative materials and seeds. Other classification is insect transmission and non-insect transmission, as most of the plant viruses are transmitted by insects. The major insect vectors are aphids, whitefly, thrips, beetles, or tree hoppers. Under non-insect transmission, you will find nematodes, plasmodiophorids, then mites, dodder, seeds, mechanical transmission, etc. The two types of interaction occurs in the host plant, that is interaction between host and the vector, and interaction between the virus and the host. Non-persistent viruses are those which are acquired by the vector after a short period of feeding, retained for some time, and are transmitted within short period, which may be from few seconds to few hours. But persistent viruses are those which multiply in the vector body and are retained in the body of such vector for longer period than that of non-persistent virus. Under these again we will find persistent propagative and persistent circulative. Persistent propagative, they are actually replicates inside the vector as well as in the host plant. But persistent circulative, they move or circulate in the insect vector, but they do not replicate inside the insect vector. Coming to the symptomatology of plant viruses, symptoms on leaves may be changes in leaf morphology like leaf rolling, leaf curling, leaf distortion, rugose, or ination. Changes in leaf color includes leaf mottling, mosaic, chlorosis, vein clearing, vein yellowing, vein banding, or leaf spots. Citrus stem pitting caused by citrus tristeza virus, then ring spot on cucurbit fruits due to some viral infection, then tuber cracking, seed mottling, then stunting and dwarfing of banana plants due to banana bunchito viruses. Then I already told you about the variegation of tulip flowers. So the symptoms expressed in almost all parts of a plant. So here I am going to explain about some of the important crop diseases caused by viruses. Chili leaf curl disease. The symptoms includes leaf curling towards the midrib, and it become deformed, stunted plant growth due to shortened internodes, and leaves greatly reduced in size, and it is transmitted by Bemisia tabaci. And mosaic disease of chili, where you will see light green and dark green patches on leaves. The insect vector is aphids, Myzus fursici. Banana bunchy top disease caused by banana bunchy top virus. The symptoms include rosette or bunchy top appearance of the banana plant, and it is transmitted by Pentalonia nigronervosa, banana black aphid. Citrus tristeza virus is one of the prime cause of citrus dieback, and it is transmitted by Toxoptera citricida, that is brown citrus aphid. Yellow vein mosaic of okra. The symptoms include alternate green and yellow patches, vein clearing and vein chlorosis, etc. And insect vector of yellow vein mosaic of okra is whitefly, that is Bemisia tabaci. Papaya leaf curl disease. It causes curling, crinkling and distortion of leaf, rolling of leaf margins inward and downward, thickening of veins. And the insect vector of this disease is whitefly, Bemisia tabaci again. Then papaya ring spot disease. The symptoms include vein clearing, puckering, then your mosaic mottling, dark green blisters, leaf distortion, which results in shoestring symptom and stunting of plants. The insect vector is Apis gossypi and Apis craxivora. Then tomato leaf curl virus. The symptoms includes the reduced internodes, giving the plant its stunted appearance. Leaves wrinkle, curl upward, giving them a cup-like appearance. The insect vector is whitefly. Then potato virus Y in potato. Leaf symptoms of potato virus Y in potato can ranges from mild to severe mosaic or mottling. Infected plants are often stunted and it is transmitted by aphids. Rice tungro disease is caused by rice tungro virus. It causes leaf distortion, discoloration, stunted growth, reduced teeler numbers and sterile or partly filled grains, and it is transmitted by leaf hoppers. Coming to the composition of a virus. It has a covering and central core. Under the covering, it includes capsid and enveloped. All viruses do not have enveloped, so that way also we have enveloped viruses and non-enveloped viruses. Then in the central core, you will find nucleic acid molecules and sometimes in some viruses, matrix protein enzymes also there. Nucleic acid is enclosed with a protein coat called a capsid, and this capsid may be surrounded by a lipoprotein layer called enveloped in case of enveloped viruses. Baltimore classification defined and classified viruses into 7 groups based on the nucleic acid, their combination, strandedness, sense and method of replication. Capsid is made up of protein coat that is called capsomere. The chemical unit of capsomeres are actually polypeptide molecules. Capsid protects nucleic acid from nucleases and other environmental factors, and capsid introduce viral nucleic acid into the host cell by the absorption mechanism. So in a virus particle, 60 to 95% is made up of protein sheet and only 5 to 40% is nucleic acid. The envelope is derived from host cell membrane, so chemical structure of envelope of viruses is similar to that of cell membrane of the host cell. Envelope viruses acquire their membrane by budding through the cell membrane, endoplasmic reticulum, Golgi apparatus or nuclear membrane. Adsorption is the first step in virus multiplication process. It occurs through random collision of viruses with host cell membrane. When a virus collides with a receptor on the surface of the cell, it adheres to the cell. In non-enveloped viruses, this adhesion is facilitated by capsid protein. As far as enveloped viruses are concerned, spike proteins play a key role in adhesion of virus on the host cell, and these spikes over the surface of the viruses are some glycoprotein molecules. So next, coming to the shapes of viruses, rod-shaped particles. they are rods or filaments, so called because of their linear array of nucleic acid and protein subunits. One important example is tobacco mosaic virus and tobacco rattle virus. Then filamentous particles, they are form of thread or filament. Important example is citrus distasia virus, potato virus Y, etc. Then isometric particle being a geometric phase system of three equal axes lying at right angle to each other. For example, Sobemovirus. Then bacilliform particles. Example is Rice-Tungro bacilliform virus. The plant virus possessing a bacilliform shape. Then membranous cuticular particles. They are envelope viruses surrounded by an outer lipid membrane. Example is tomato spotted wilt virus under Tospovirus genus. Then thin flexuous multipartite virus. The shape is full of bands and curves. Multipartite viruses appear mostly, they are restricted to plants and fungi. So coming to the fundamental types of symmetry of plant viruses. The capsid is composed of a number of subunits called capsomere, and these capsomeres are arranged symmetrically to form a shield around the nucleic acid core. Based on this arrangement of capsomere, we find actually three types of symmetry in plant viruses, or in viruses actually. Icosahedral, that is 20-sided. It is also called polyhedral or spherical symmetry. Then helical symmetry is also called cylindrical or rod-like symmetry. Then complex or binal symmetry. In helical symmetry, the monomers and nucleic acids are arranged in a helix around a spiral tube. A virus that has its capsid shaped into a filamentous or rod-shaped structure is of this symmetry. Then icosahedral symmetry, where a virus consisting of identical subunits that make up equilateral triangles arranged in a symmetrical fashion. Viruses employ the icosahedral symmetry because it is the most efficient way to enclose a space. It is a regular polyhedron with 20 equilateral triangular facets and 12 intersecting points or corners. The capsid appears spherical when viewed at low power in the electron microscope. These are most common than helical symmetry. In complex viruses, capsid symmetry is neither icosahedral nor helical. Binal symmetry possesses a combination of both icosahedral head and helical tail. For example, is bacteriophage. In one type of complex symmetry, there is a multi-layered wall around the nucleic acid. The protomers are all identical in structure but irregular in arrangement. For example, is pox virus. Coming to the replication of plant viruses, there are actually 6 steps: attachment, entry, replication, biosynthesis, assembly, and release. The process of multiplication in viruses is generally referred to as replication. Here the viral protein and the nucleic acid components are reproduced within the susceptible host cell. Specificity of viruses depends on the type of receptor over the host cell surface, which helps in the attachment of viruses to the host cell. Then once inside the cell, instruction to the nucleic acid to transcribe to RNA. That is the replication process. And then protein building machinery of the host cell then translate its instruction into protein. That is the component of a new virus. Some viruses replicate inside the nucleus and virus move out into the cytoplasm. For example, colimovirus, Gemini viruses, Potex virus, or Tobemovirus. But some viruses replicate in the cytoplasm, for example, bromovirus, potivirus, nipoviruses, or comoviruses. Uh, Today we have discussed about basics in plant virology, which includes introduction and importance, history, the pioneer history, structure, composition, shapes, symmetry, replication, etc. in a nutshell. Hope all the students will be benefited. I'm concluding this lecture here with a message: sit like a rock and work like a clock. Today's preparation determines tomorrow's achievement. Thank you.